Hello, in this video I will explain about glycogenolysis, which is one of our metabolism system in our body. So the first of all is if we break down the terms glycogenolysis, we get a glycogen in places. Basically, it's mean breaking down of glycogen. So simply, glycogenolysis is a process of breakdown of glycogen into glucose. But before we understanding glycogenolysis, we should ask ourselves 3 points. The first is where does it take place? Second is when does it take place? And the last is how does it take place? So let me answer those 3 questions point by point. And now, seat for glycogenolysis is muscle and the liver. So that's answer of the first question. Where does it take place? But why it takes place in the muscle and in the liver? Simply it's because all of enzymes, it just like glycogen, phosphorylase, differentiating enzyme, and phosphoglucosamidase. These enzymes, which are essential for glycogenolysis, is present in our liver and our muscle. And also the substrate, its main glycogen, which is broken down into glucose, is actually stored in our liver and our muscle. So that is why this primary set of glycogenolysis. So then we ask, when it takes place? It takes place in various situations when our body requires additional glucose for energy. Here are some conditions when glycogenolysis occurs. The first is during physical activity, second is between meals or during fasting, and the last is during periods of stress or low blood sugar. So, when this situation occurs, our body needs an energy, and to obtain that energy, glycogen will be broken down into glucose, which is the process of glycogenolysis will take place. Now, how does it take place? So, first of all, we have a glycogen, which is the starting material of a glycogenolysis, consists of 18 molecules of glucose, which are branched. The glucose which are connected in a chain are connected by alpha 1,4 glucosidic bonds, but the bonds formed at the branch junctions are connected by alpha 1,6 glucosidic bonds. Now, glycogen in the presence of the enzymes glycogen phosphorylase gets converted into glucose 1-phosphate. This enzyme works until 4 residues of glucose are left on the other side. 4 residues of glucose when left on the other side form limit dextrin. Limit dextrin, which is formed now, cannot be further degraded by phosphorylase, and the branching of glycogen are divided by the branching enzyme. This the branching enzyme. And now, first of all, the debranching enzyme, the fragment of glucose molecule which contains 3 molecules of glucose is transferred to these 3 molecules is transferred to another chain, and over here the branching enzyme shows transverse activity. Now, the resulting glycogen molecule come under the action of the branching enzyme, where alpha 1,6 glucosidics breaks the alpha 1,6 bond, and single glucose molecule are released. This is a single glucose molecule. So now, the remaining molecule of glycogen. Oh wait, this is now the remaining molecule of glycogen is again available for the action of phosphorylase and gets converted into glucose 1-phosphate. This converted into glucose 1-phosphate. And then glucose 1-phosphate in the presence of enzyme phosphoglucomutase get converted into glucose 1-phosphate. This glucose 1-phosphate is further used in the glycolysis process, but in our liver, glucose 6-phosphate under the presence of the enzyme glucose 6-phosphate get converted into glucose. So there is process of glycogen, which is glycogenolysis, which is a glycogen convert into breaking down into glucose.